$14.8 million US dollar per year. The productions of banana, like other crops, is often limited by several constraints. In general, for the past 3 years, the trend of planted areas of banana in Malaysia has been declining from 34,000 over hectares to 26,000 hectares, as you can see in the graph 1. Similarly to the trend of banana production from 350,000 over ton metric in 2017 to 325,000 over ton metric in 2019. One of the contributing factors to the deterioration of these productions is the major disease infection. We have two major diseases in Malaysia. One is Fusarium wilt or Panama disease, and secondly, bacterial wilt disease, or known as moko and blood disease. Apart from that, we have three minor diseases. One, Sigatoka leaf spot. We have both black and yellow Sigatoka, banana bunchy top virus, and banana streak virus. Now we are at 3.0, the status of Fusarium and bacterial wilt diseases of banana in Malaysia. For this, I'm going to touch a bit on the history and the current updates. We have been living with Fusarium wilt disease since a long time ago, and however, the incidence of Fusarium wilt disease has further dampened the banana industry when the emergence of the broad-spectrum strain of Fusarium oxysporum from a specialist cubense named tropical racefor, or known as f.sp.cubense in the 1980s. Shortly after Malaysia ventured into the cultivations of Cavendish banana. The disease incidence has been reported for up to 70% in the field. Presently, bacterial wilt disease is considered the most damaging disease of banana and lists as a top 10 invasive alien species in Malaysia. The infection could cause up to 100% when it's outbreak. The bacterium that cause bacterial wilt disease are Ralstonia solanacearum race 2, Biovar 1, or we call it as a moko, and Ralstonia syzygii subspecies celebesensi, or blood disease. The first incident disease was detected in 2007, has resulted in remarkable yield losses and increase of banana importation value from 0.37 million ringgit Malaysia in 2007 to 7.9 million ringgit Malaysia in 2012. Presently, no effective chemical to control the diseases. Almost all popular banana cultivars are susceptible to the diseases. Now we are at the R&D status of biological control approaches to control bacterial wilt and Fusarium wilt diseases. Biological control approach have received increasing interest due to environmental friendly. This study focuses on obtaining bacteriophage, the naturally occurs viruses of bacteria and streptomyces species, a gram-positive bacterium with the filamentous-like fungi. If you can see on your right side here, yeah, this is the images of bacteriophage. Yeah, this is the TM image that we clearly can see the head and the tail. Yeah, and here the drawing of bacteriophage, which consists of single or double-stranded DNA or RNA genome encapsulated inside a protein or lipoprotein coat. From the down here, it's a streptomyces species, a gram-positive bacteria with a filamentous-like fungi. Bacteriophage tend to persist in high numbers in any environment as long as the host, which means bacteria, is present. Generally, phage have a narrow host range, which is very good because it's very specific to the pathogen. Yeah, typically being limited to strains within a particular species of bacteria, for example, Xanthomonas, Pseudomonas, Avinia, and Ralstonia species. Streptomyces species has been explored as a biological control agent for controlling various plant diseases. For example, bacterial panicle blight due to Buclodoria grume on rice, and bacterial leaf blight caused by Xanthomonas oryzae. Therefore, Mardi took an initiative. We proposed to isolate and evaluate the potential isolates of H and Streptomyces species as biological control agents against bacterial wilt and Fusarium wilt diseases. This is to show you the process of isolations and evaluations of lactic bacteriophage in vitro and in the glasshouse. First, isolations of bacteriophage from cytostem, soil, and fruits of infected banana in infected field. Second, the presence of lactic bacteriophage was evaluated from the sample collected using double-layer agar method. In this, Ralstonia syzygii subspecies celebesensis used as the host. To clarify the bacteriophage into morphology-specific group, phage particles were examined under transmission electron microscope. From 20 samples, only 2 were identified as bacteriophage, which belonging to the Podoviridae family. Then those phage were subjected to evaluate in the glasshouse on 2 months old of banana plants by several treatments. This is the disease severity scoring index that the team used for the treated banana plant. The summary of methodology on microencapsulated of Streptomyces species to control FOC. First, sample collections and isolation of Streptomyces species from banana crops environment. Then we go through process of in vitro screening of Streptomyces species against FOC. Then we prepare liquid formulations of Streptomyces species in actinomycin broth. We also formulate the Streptomyces species in microencapsulated using calcium arginate beads. And all these are subjected to evaluations of different isolate of Streptomyces species against FOC in a glass house, as well as in the FOC hotspot field in one of the banana field in Mardi station. These are some of the results to show you. As you can see in the graph 2 here, in vitro evaluations of single and cocktail phage against Ralstonia syzygii subspecies celebesensis in CPG liquid medium based on absorbent value. The bacterial suspensions of Ralstonia syzygii infected with individual phage number X9105 and X513 and cocktail which contain X9105 plus X513 began to show infectivity effect after 3 hours of incubation period compared to the bacterial suspensions of Ralstonia syzygii subspecies celebesensis without phage as a control. The cocktail phage of X9105 plus X513 shows the highest infectivity effects against Ralstonia syzygii subspecies celebensis in vitro as compared with other treatments including control. Then to the graph number 3 here on your right side, implanted assay of phage treatment on 2 months old banana plants. We found out that T7 cocktail phage of X9105 plus X513 at a concentration of 10 to the power of 8 plug forming unit per male plus Ralstonia syzygii subspecies celebensis at 10 to the power of at similar concentration as a soil drenching technique shows the lesser percentage of disease severity at 23.3% as compared to positive control. Here, T3 at 63% in the glasshouse evaluation. This is to show you a picture at the very bottom here, the visual symptoms on the treated banana plants at 30 days after phage application, where the number 1 is the negative control, number 2 is the positive control, and number 3 here showing 3 plants with phage treatment as a cocktail as a soil drenching method. These are the results for Streptomyces species. Yeah. Table 2 here shows effects of different Streptomyces species isolates towards the growth of FOC in dual culture test in vitro. In vitro evaluation found that Streptomyces species designed as 12 gave significantly the highest ability to suppress FOC as compared with other treatments including control. Whereas for in glasshouse evaluation, we found that banana seedlings pre-inoculated with FOC, then treated with Streptomyces species designed as S1, S5, S7, S12 were recovered and survived throughout the 21 days observation period. While untreated banana plants or controlled died after 7 days treated with FOC. These are the treated banana plant with FOC or positive control. Yeah, this to show you the plant recovered after pre-inoculated with FOC. This is the early symptoms of plants, banana plants infected with FOC. Then number 3 here in preliminary field evaluation found that T2 Streptomyces S5 shows the highest plant survival percentage at 96.3% compared to other treatment. This is the view of banana plants in the field. Yeah. This is the treated with Streptomyces species, and this one is the control plant. When we go for conclusions and way forward, first, bacterial wilt and fusarium wilt diseases of banana could be controlled using the biological approaches with selected microorganisms as biological agents in a suitable formulation. The bacteriophage cocktail isolate X9105 plus X513 is one of the potentials biological control for bacterial wilt disease that will be formulated and further tested in the bacterial wilt disease hotspot field. The microencapsulated of Streptomyces species designed as 5 is the potential of biological control for FOC-TR4 that will be upscaling before commercialization. With that, I thank all of you for your all attention.
Okay, thank you very much, Dr. Rozeita. So I would like to invite the floor for if you have any questions from the floor. So we've got like 2 more minutes. So perhaps I could give you one question, yeah, Dr. Rozeita.
Yes.
Could you go to the treatment, the graph showing different treatments when you put Ralstonia syzygii.
It's quite difficult to pronounce them.
Yeah.
Yeah.
So I just want to know what's the difference between the treatment because I think you've got up to like 10, 12 treatments.
Yeah.
And
I thought
7 was good.
Okay, thank you, Dr. Sharifah, for the very good question. I thought so. Someone will ask me about those graph, yeah. Because the time is so limited, so during the presentation, I haven't got any chance to explain detail, yeah. So if you can see, if you can still remember, either someone can show again the graph, yeah. So the team, the researcher tried to use several technique to treat the plant, either using drenching technique, and then using injection as well. So and then either a single for each of the phage, and cocktail means we combine the two phage together. So okay. So if you can see, T1, T2 is a negative control, and T3, which mean the positive control, the bacteria, the bacteria, they just drench into the soil, drenching. And T4 by injection, the phage, they just inject, not the phage, the bacterial wilt disease, they inject into the plant, and it looks that by injection, the disease quite a little bit higher as compared to the drenching technique. And then T5 is the phage, 9105, and together with bacterial wilt disease by drenching. And T6, phage X513, together with bacterial wilt disease as a drenching. T5 and T6 and T7 all drenching. T7 is a cocktail of both phage together with bacterial wilt disease, and looks like the T7, the cocktail together with the bacterial disease, more or less disease severity as compared to the T8, T9, and T10, where this group or this cluster, they infect the plant using injection. So the researcher found out that when we inject the plant, inoculate the plant using injection, we inject the phage into the plant, then the phage could not survive as better as drenching, because as we know that virus is very sensitive to UV and heat. So when we do drenching, it looks much more better. The phage survive better as compared to the injection.
Okay. All right. Thank you, Dr. Rozeita. So in the interest of time, I would like to just proceed with the next presenter. Thank you, Dr. Rozeita.
Yeah, you're welcome. You're most welcome.
Yeah.